Dear colleagues, first of all, I would like to thank the organizers for inviting me to come both to the Columbian College or meeting of rheumatology and also Columbian Congress of Pediatric Rheumatology. And I would like to apologize to all of you for not being able to come to this meeting in person. Also, I would like to thank Ms. Jamie Diaz for trying very hard to bring me to this meeting. Unfortunately, my travel was not approved due to some unforeseen circumstances. So I was asked to talk about the recently identified and characterized auto-inflammatory disease, which goes by the acronym of VEXAS syndrome. So before I start talking about the VEXAS syndrome, I would like just to briefly remind you about auto-inflammatory diseases. Basically, systemic auto-inflammatory diseases are either inherited or acquired diseases of the innate immune system that result from the antigen-independent hyperactivation of various inflammatory signaling pathways such as TNF, IL-1, or interferon signaling pathway. These patients present with excessive either systemic or organ-specific inflammation, as you will see in my slides. And what it means also is that inflammation in these diseases is predominantly mediated by myeloid cells, namely monocytes, macrophages, and neutrophils. So genetics of monogenic systemic auto-inflammatory diseases, it's very complex. We've got some autosomal recessively inherited diseases, dominantly inherited diseases, a few X-linked diseases. There is also evidence for digenic inheritance, oligogenic inheritance, polygenic inheritance. And in more recent years, there has been increasing evidence for the contribution of somatic mutations to the pathogenesis of systemic auto-inflammatory diseases, and one of these diseases is actually a VEXAS syndrome. So basically, the difference between germline and somatic mutations is in that somatic mutations arise in post-zygotic stage, and their effect is influenced by the time of their occurrence during development and the affected cell type. So patients that have these somatic mutations early in post-zygotic stage typically present with early onset symptoms, while patients that have mutations occurring later in development present with late onset symptoms and sometimes milder phenotypes. In patients with systemic auto-inflammatory diseases, mutations are found in genes that have been previously linked to diseases via germline pathogenic mutations, except for the VEXAS syndrome. So the first time these somatic mutations were reported in patients with auto-inflammatory diseases was almost 20 years ago, with identification somatic mutations in patients that have so-called cryopyrinopathies. And these somatic mutations are predominantly found in myeloid cells. And studies also show that these somatic mutations often arise at the residues where germline mutations are likely incompatible with life. And also the frequency of this mutant allele can increase over time and can lead to progressively more severe clinical presentation. This table from a recent review actually showed several auto-inflammatory diseases that have been shown to be linked to somatic mutations in respective genes. So VEXAS syndrome has been described and reported only a couple of years ago. And basically, it's caused by somatic mutations in the X-linked gene, UBA1, which encodes E1 ubiquitin activating enzyme, which is required for the post-translational modifications by ubiquitination of all cellular proteins. So essentially what it means is that this enzyme is critical for the cellular functions, and that the germline mutations in this enzyme are likely incompatible with life. So the key features of VEXAS disease are fevers, neutrophilic dermatosis, pulmonary infiltrations, and other features that I will show to you in the upcoming slide. So here you can see the summary of clinical features in patients with VEXAS syndrome. In the first initial cohort of patients that included 25 patients referred to NIH, And then again, a summary of these clinical features in expanded cohort of patients of 733 patients, essentially. So what you can see here is that most of these patients present with fever, with neutrophilic dermatosis, with lung disease, pulmonary infiltrates, with relapsing chondritis, and also about a third of them present also with thromboembolism. What this figure showed here is that there are other features that are also observed in this disease, such as, for example, CNS inflammation in the form of eye inflammation, sensory neuronal hearing loss. Then, in addition to pulmonary infiltrates, some of these patients present with pleural effusion and myocarditis, bone marrow dysplasia that I will discuss in the upcoming slide, with inflammatory arthritis, and some of these patients actually also had arterial strokes. So frequently reported hematological changes include microcytic anemia, which is shown here, cytoplasmic vacuoles in myeloid and erythroid precursors, although sometimes these vacuoles are also seen in circulating neutrophils in peripheral blood. Here you can see actually evidence for these cytoplasmic vacuoles in myeloid cells. Then many of these patients present with bone marrow hypercellularity, and they present with varying degrees of bone marrow dysplasia. However, they don't typically progress to develop leukemia. As I said, some of these patients present with myelodysplastic syndrome. Some of them also have propensity to develop macrophage activation syndrome, or HLH. And what we like to say to rheumatologists is, if you have a patient with severe systemic inflammation and bone marrow dysplasia, think of vexus. To oncologists, we like to say, if you have a patient with myelodysplastic syndrome and severe systemic inflammation, or essentially inflammatory MDS, again, you should think about vexus. So because some of these patients present with MDS, a recent study in France showed that at least patients that have vexus and associated MDS, some of them were responsive to treatment with hypomethylating antineoplastic drug as a citizen. So that's something that could be considered in the treatment of these patients. So what you can see here on this positron emission tomography is that essentially patients with vexus syndrome have very, very active bone marrow. So this is the table that I borrowed from this original paper that was published a couple of years ago in New England Journal of Medicine. What you can see here that in this initial cohort of 25 patients, all these patients were males because this is an X-linked disease. The median age of onset is about 65 years, and mortality was very high. It was about 40%. And these patients died from disease-related causes, such as infections, respiratory failure, or progressive anemia. And some of them also died of complications of the treatment, because by the time that we were seen at NIH, most of them were treated with high dose of steroids. What you can also see here is that all the patients in this initial cohort carried missense mutations at one particular residue, methionine 41, that is important for the translation of the isoform of this protein. So the vexus-associated somatic mutations originate in multipotent hematopoietic stem cells, and they are found at a variable frequency in peripheral blood myeloid cells, but they are not found in T cells, B cells, buccal cells, or fibroblasts. And so the hypothesis why they are not actually found in cells of the adaptive immune system could be that either these mutations are somehow advantageous to myeloid cells, or alternative hypothesis is that basically they are just toxic to the T and B cells. And I personally believe that these mutations are toxic to the cells of the adaptive immune system. The frequency of mutant allele is very high. It's anywhere from 40% to 90 or even 100%. Sometimes I cannot even see the wild type allele in these patients, which essentially means that molecular diagnostic testing can be done by simple Sanger sequencing. In patients that have a lower variant allele frequency, we need to use other methods such as next-gen sequencing or digital droplet PCR. Interestingly, the variant allele fraction in peripheral blood DNA does not correlate with disease onset or severity, and it remains to be seen whether there is a better correlation between the variant allele fraction and found in DNA samples that are extracted from bone marrow samples of these patients. So basically this enzyme is encoded by the gene UBA1, and this gene or this enzyme actually exists in two isoforms. It exists in the nuclear isoform, which is expressed in all cells, and the cytoplasmic isoform, which is expressed exclusively in hematopoietic cells. So what you see in wild type, or basically cells of a healthy individual, you see this nuclear isoform, which is shown here with these purple dots, and green dots represent the cytoplasmic isoform. What's happening with patients that have VEXAS, they carry these missense mutations that affect the translation of the cytoplasmic isoform of this protein. So essentially, they cannot make this isoform, which is shown here with green dots. Rather, they start translation at the next methionine downstream in this gene, and they make this isoform that we named isoform C, which is catalytic death. So basically, they lack functional UBA1 enzyme in their hematopoietic cells. So what is seen also in monocytes of these patients is that they have decreased level of polyubiquinated proteins and also increased level of free ubiquitin, but only in monocytes. You don't see this cellular phenotype in their T cells. So you can see here that basically they have this ubiquitously expressed isoform A. They have very little, expression of the isoform B, and then they have a high expression of this catalytic death isoform C. So the loss of ubiquitination and accumulation of proteins that are not properly ubiquinated in monocytes or myeloid cells may activate cellular stress responses, which will lead to the upregulation of UPR, unfolded protein responses, dysregulation in the autophagy. Ultimately, that's going to trigger inflammatory responses in mutated cells, and that's why these patients have high levels of many inflammatory cytokines in their peripheral blood. So some genotype-phenotype correlations show that the survival length of patients with VEXAS syndrome is dependent on the mutation. So basically, the median survival length in VEXAS syndrome is about 20 years. However, patients that carry this particular mutation, methionine 41-valent, have a shorter survival length, and that can be explained with the observation that basically this mutation is associated with a lower production of this isoform B compared to the other two missense mutations. The current estimate of mortality of patients with VEXAS is a little bit lower than it was initially estimated, and it is about 25%. So patients with VEXAS share a lot of clinical features with patients that have been diagnosed with other rheumatological diseases. So what you can see here is that, again, from this initial paper, that about 60% of these patients were clinically diagnosed with relapsing polychondritis, about a third of them with Sweet syndrome, about 25% of them with myelodysplastic syndrome or multiple myeloma, polyarteritis nodosa, or other types of vasculitis. Nevertheless, a recent study from NIH basically looked for them in a cohort of patients that are clinically diagnosed with relapsing polychondritis. That cohort included about 90 patients with relapsing polychondritis, and basically they were sequenced for mutations in this gene, and only about 8% of these patients were positive for mutations in this gene, UBA1. So the three features that really distinguish patients with VEXAS and other patients with relapsing polychondritis are male sex, mean corpuscular volume higher than 100, and the platelet count which is lower than 200 and 1,000. And basically that really differentiates patients with VEXAS RP from patients with relapsing polychondritis with 100% sensitivity and 96% of specificity. In addition, there are some other clinical features that can basically point that a patient that presents with relapsing polychondritis may have vexus, such is that these patients have leukocytoplastic vasculitis, and a lot of them have neutrophilic dermatosis, which are not typically seen in patients with classical relapsing polychondritis. Unfortunately, the mortality of patients with vexus RP is higher than the mortality of patients with classical RP. Few words about the treatment of these patients. So as I previously mentioned, mortality is very high, and it's absolutely critical that these patients are diagnosed early in the disease course, that their diagnosis is confirmed by molecular diagnostics, because they are more likely to respond to therapies even with cytokine inhibitors. So although the patients that were seen at NIH, by the time that were seen in our clinic, basically most of them were just had a severe damage in bone marrow, and they had irreversible damages in their bone marrow, and they didn't respond to any cytokine inhibitors. So what these studies showed is that basically patients with VEXAS syndrome may respond to treatment with JAK inhibitors. And what this study showed is that the clinical response and also biological response was achieved in about 50% of patients that were treated with JAK inhibitors 1 month after the treatment, and that basically this clinical response was better in patients that were treated with ruxolitinib compared to patients that were treated with other JAK inhibitors. That also correlated with the reduction in the dose of prednisone that was used in patients treated with ruxolitinib compared to patients that were treated with other JAK inhibitors, and also correlated with the increase or rise in the hemoglobin levels. Again, this increase was more pronounced in patients that were treated with ruxolitinib compared to patients that were treated with other JAK inhibitors. In terms of side effects, the most common side effects of treatment with JAK inhibitors, as expected, were infections, some thromboembolic events, and 3 of these patients died. Actually, 1 of them died from infection, was treated with tocilizumab, 1 from colon cancer progression, and 1 of them died, but the cause of death was not disclosed. So this study also showed that despite the good clinical response, the mutant clone, a mutant UBA1 clone, essentially progressed, despite the fact that these 2 patients had a good clinical response, as shown here with the relatively controlled levels of acute phase reactants. And basically, eventually, these UBA mutated clones outcompeted the other 2 mutations, namely deletion of 7q chromosome and JAK2 mutant clone in these 2 patients. So essentially what this study showed is that ruxolitinib has mainly a suspensive effect, or basically can suppress inflammation in these patients, but cannot cure patients with VEXAS syndrome. And then here is another study that was just recently published, which showed that ultimately the only successive therapy could be hematopoietic stem cell transplantation, providing that these patients are diagnosed earlier in the course of disease and that they have a good match donor. So what this study showed is that 5 patients, so this is a retrospective study of 6 patients that actually had bone marrow transplantations before they were diagnosed with VEXAS. So these patients, the 6 patients were transplanted for the reason that they had a severe refractory inflammatory disease or myelodysplastic syndrome. And what you can see here is that in this transplantation, they used both related and unrelated donor with a good identical match. And they observed some complications, such as infections complications, which were observed in 4 out of 6 patients. GVHD was observed also in few of these patients. Nevertheless, 5 of these patients are still alive. So this study was recently published, and basically this study analyzed the exome data from 160,000 of participants from the Geisinger Community Health Initiative, and in this huge cohort of actually patients, 11 individuals were found to have clinical manifestation consistent with VEXAS syndrome, and they were found to carry pathogenic or likely pathogenic somatic variants in the UBA1 gene. Interestingly, 2 of them were females, and these patients or women actually with VEXAS They are very rare, but it's possible. There is no reason why they cannot acquire somatic mutation in this gene, but these patients will either have monosomy of X chromosome or the phenotype in women can be explained by skewed X inactivation. The estimated disease prevalence in individuals older than 50 years is 1 in 4,000 men and 1 in 26,000 women. In conclusion, I would like to say that identification of pathogenic, germline, and somatic variants has contributed to better understanding of molecular pathways driving inflammation. Identification of disease-associated mutations enables a patient-tailored therapy. And finally, somatic mutations should be considered in patients with various adult-onset immunological disorders, including patients with late-onset autoimmune diseases such as RA and lupus. I would like to thank you for your attention, and I'm looking forward to seeing you tomorrow online, and I will be happy to answer to your questions.